All right. So, welcome everyone to the first webinar of IISA of this year. I'm Hia Banerjee. I'm the president-elect for IISA. So I really want to welcome all of you, and I hope like everybody can see us and hear us. So we just want to mention, this is the first one, and we actually plan to run it almost every month. So we will keep you guys updated on that. But since this is the first one, I actually want to talk a little bit about IISA. Maybe you guys are already aware of it. It is International Indian Statistical Association. This prestigious society has more than 2,500 members worldwide. Our mission is to promote education, research, and the application of statistics and probability across the globe through the collaboration and shared learning. So we encourage all of you to consider joining IISA if you haven't done it yet. And please participate also, in our different initiatives. So one of the initiative I want to mention that we are conducting our annual conference this year in India, in Kerala, in December. So please, go to our website and you will have all the updates about our organization. So saying that, I also want to take this opportunity to introduce you to the webinar committee who actually made this event possible. So please join me, recognizing Himal Malik. He's the assistant professor at Cornell University. Piali Boshak, who will be moderating today's session. She's a biostatistician at Merck. Jyotish Kodatto, he's an assistant professor at Virginia Tech. And Kaushik Jana, who is also an assistant professor at Ahmedabad University. So please give them a well-deserved round of applause. Thanks a lot. But before I introduce to our speaker today, there are some house rules. This webinar is being recorded, and please use your Q&A for your questions or comments. And there will be a feedback form which will be sent to after we finish the webinar. We really appreciate your input to help us to improve our future webinars. So please, fill that up. So again, thanks a lot for joining. But now it is our time to introduce with the speaker. And today's speaker is Dr. Devan Mehrotra, who is the Vice President of Biostatistics at Merck Research Laboratories. He has made significant contribution to the research and development and regulatory approval of medical drugs, vaccines across a broad spectrum of the therapeutic areas. So he's also an adjunct professor at the University of Pennsylvania. Besides that, he also serves as subject matter expert for the Bill and Melinda Gates Foundation, U.S. National Academy of Science, and also Coalition for Epidemic Preparedness Innovations for COVID-19 vaccine development. So it is our immense pleasure to have you, Devan, here for this year's webinar. And I'm sure most of the IISA members are aware of you, but if you want to say maybe 1-2 lines about your involvement with IISA, that will be great. And after that, the floor is yours. Thank you.
Thank you so much, Hia and Himal, for this very kind invitation. I'm so happy to be here. IISA is like a family to me. I've been involved in some shape or form with IISA for many years, it's always a pleasure to actually attend the in-person meetings. I've gone to a couple of meetings in India. I've attended the meetings when they've taken place in the US, and really, I've also played really a very small but hopefully meaningful role in helping them identify future leaders, including senior leaders as presidents and so on, just to identify some candidates. So again, thank you so much for the kind invitation. It's my pleasure today to then talk about really an interesting topic. Is inadequate risk stratification diluting hazard ratio estimates in randomized clinical trials? So I'll just ask that if your line is unmuted, just so that everyone can listen in without interruption, maybe you can just keep your line muted. And I do understand and appreciate that we're in the middle of the T20 Cricket World Cup. And so maybe some people will be multitasking, looking at the screen and then looking at the TV screen. And that's okay, because I've been following with great interest some of the exciting matches myself. But but hopefully this is sufficiently interesting. And so the short answer to the question in the title is yes. So if you want to go back to the cricket match, that's okay. But if you're interested in knowing why the answer is yes, then I will explain this in the next maybe 20, 30 minutes, and hopefully there should be plenty of time for Q&A. Let me put this in presentation mode so it's easier for me to look at this on my screen. And I will make sure just a single screen. Right. So this is joint work with my colleague, Rachel Marceau West at Merck. She joined us from North Carolina University about 7 years ago. Okay, so interestingly, the top half of my screen is being chopped up, but that's okay. So the outline is fairly straightforward. If you're not familiar with survival analysis in randomized clinical trials, I'll just give you a very quick introduction to survival analysis and really talk about the log rank test, which is very commonly used. And in particular, when people talk about the log rank test, they often refer to as the unstratified log rank test, although they may not explicitly mention that. We then make our way to this new approach called 5-star, which I will motivate and explain, and importantly, apply that to three fairly large cardiovascular outcomes trials, where the sample sizes in these clinical trials range anywhere from 5 to 30,000 people, and then summarize and then open it up for Q&A. So the log rank test, again, is very commonly used in clinical trials. And so we are envisioning a randomized clinical trial. There are two treatment arms, A and B. A is the test treatment, the new treatment, and B is the old or the control treatment. We are looking at survival times until an event of interest. And the event of interest could be, it could be cancer progression, it could be time to somebody having a heart attack, it could be time for cardiovascular death. So depending on what the drugs that are being studied, what they're trying to prevent or delay, the context will determine what exactly the survival time is. So in general, I'm just going to use words, the expression survival time. It need not be time to death, but I'll just use the word survival time. And it's fairly well known in the survival time literature that if the underlying hazard functions associated with the corresponding survival function, if those hazard functions are proportional, and what do we mean by a hazard function? What is a hazard? So a hazard, very simply put, is at any given time in the trial, let's say at 10 months, if a certain fraction of the people have still not had the event, then and if you're measuring event times in days, then what proportion of those who are still event free on a given day, the next day, what will be that survival fraction? What is the percent reduction in that survival fraction from one day to the next? That's referred to as the hazard function. And if the treatment hazard functions are proportional to each other, then it's well known that the log rank test is most powerful. And one way to visualize if that assumption is reasonable is to plot the log of the survival function over time. So if you have Kaplan-Meier curves, which are typically used, if you plot the log of the estimated survival using Kaplan-Meier curves for treatment A and B, and if they are proportional, if that's relatively constant over time, then the pH assumption seems to be reasonably satisfied by the data. And in truth, if they are, then that common ratio is referred to as hazard ratio. And shown here in the left is really what is the log rank statistic, how is it calculated. So this was, of course, proposed back in 1960s, and then David Cox, in his famous 1972 paper, he then showed that this test, the Mantel log rank test, is the score test from the Cox proportional hazards model. So this is just a very quick introduction to survival analysis and the log rank test. And what's really interesting is that in the clinical literature, and I'm showing you some among many examples in the clinical literature on how is the data reported? How are the results reported in these trials? So typically you will see these Kaplan-Meier curves, and then you will see hazard ratio, point estimate, and confidence interval being reported, as well as a p-value from the log rank test. And these are all examples where in the clinical trial protocol and as well as in the publication, these are p-values from the unstratified log rank test. So no adjustment was made for risk stratification, and I'll explain that in a few minutes. So which is quite surprising because this is actually not a good practice. There should be risk stratification, which I'll explain very soon. But again, in these leading medical journals, JAMA, Journal of the American Medical Association, JCO, Journal of Clinical Oncology, the New England Journal of Medicine in the bottom left, and then the Annals of Oncology, very prestigious journals. And what we are finding, to our surprise, is that in several cases, the unstratified Cox model or the unstratified log rank test is still being used. So why is that a concern? And the concern is explained using this toy example that I have here on this slide. So let's look at the panel on the left. So suppose in truth, in our clinical trial, there are two types of patients. Let's call them higher risk, shown in red on the bottom, and then the lower risk in blue on the top in the left panel. So the lower risk patients are the lucky patients. Regardless of whether they get randomized to treatment A or B, they will tend to live longer. They will have longer survival. The unlucky ones are the red patients who, regardless of whether they get treatment A or B, are going to be having shorter survival, and if the endpoint is death, they're going to be dying faster. So what's interesting in this scenario is that clearly there are two very different types of patients in this imagined clinical trial. And suppose in truth, for both the red and the blue patients, the new treatment relative to the old treatment, meaning the test treatment relative to the control treatment, it does improve survival. So the survival curve, so the higher the survival curve, the better, because you want longer survival. And so it's clear from this, this sort of toy example that for both the red and the blue patients, the test treatment has a better survival function than the control treatment. And in this case, because this is just a simulated scenario for teaching purposes, we know as the simulators that the truth is that the hazard ratio is constant over time, and the hazard ratio is 0.7 for both the lower and the higher risk patients. So if you were to get a single data set from this imagined scenario in a clinical trial, and if you were to do an analysis and report a single hazard ratio, it would be very reassuring if the hazard ratio that was reported is close to 0.7, because that would be a representative hazard ratio for the blue and the red patients. And if you imagine some kind of an average hazard ratio for the blue and the red patients, then in this case, the 0.7 would be perfect. But suppose we don't know that there is a mixture of higher and lower risk patients, or you choose to ignore that. And you do what is often done, and we've seen examples on the previous slide, where you see these overall estimated survival curves using the Kaplan-Meier approach, and you see the just a single set of curves, one for the test treatment, one for the control, and there is no separation into risk categories. You just see an overall curve here. And what is the consequence of this? So the consequence of just looking at an overall curve, ignoring the underlying risk levels, is you will end up with an estimated hazard function over time that is not constant. So as shown here, what we're calling the risk unstratified hazard ratio, this is when you get a single data set from a clinical trial, you will be seeing an estimate of the hazard function over time that looks approximately like this with a large enough sample size. And that's very confusing because it's not constant over time. And so the hazard ratio that you report will now becomes dependent on what is the average follow-up in that trial. which is a very unsatisfactory and hard to interpret number. And importantly, what we see here is that the hazard ratio will be closer to 1. So this is what we mean by a dilution of the true hazard ratio. So in truth, the true hazard ratio is 0.7. And what we mean by the risk stratified hazard ratio is at every time point, if you have the hazard ratio for the red and for the blue patients, and in this case, it's a 50-50 mix, conceptually, if it's just an average of those two, in truth, that is going to be constant over time. And like I said, you ought to be reporting a hazard ratio of 0.7 at the end of the trial. So this is what we mean by a dilution, and a failure to risk stratify can get you in trouble. And we've seen examples on the previous slide where the analyses are not stratifying for risk. They're not adjusting for baseline risk. Okay, so what do we do about it? Well, we start out with a recognition, and we've spoken to many clinicians across very different disease areas, oncology, cardiovascular, neuroscience, immunology, and so on. And they all agree that the patient population that enter these clinical trials, and indeed the patient population for which the experimental treatment is being investigated, hopefully it's safe and effective, and someday it will be approved. And it's going to be given to a patient population that is a heterogeneous mixture of what we call higher risk patients, moderate risk patients, and lower risk patients in concept. So if you try to do statistical inference directly on the overall population without separating them out into conceptually high, medium, low risk, then you suffer the consequence of potential treatment effect dilution, where those hazard ratios, in truth, they may be well below 1, but they are being moved closer to 1 in this biased manner. So really, we thought about this and we said, wow, this is a very simple approach that we could be using. And so what we could do is we could take the overall population before treatment unblinding. So we could pre-specify a treatment blinded algorithm. And using that algorithm, it would take the overall risk heterogeneous population, and it would separate out these patients into these risk strata, what we call risk strata. Once that has been accomplished by this treatment blinded pre-specified algorithm, now the algorithm and the analysis can be unblinded. And now for each of these formed risk strata, go ahead and control and compare treatment A to treatment B. You will get a hazard ratio, or more strictly speaking, a log hazard ratio. And you basically take an average across these for overall inference. So we call this method 5-star because it has 5 steps that are described in this box below. And I'm going to walk through a hypothetical example first to explain how this works. And then, most importantly, we will apply this to real data sets to illustrate the utility of this approach. Okay, so let's walk through a toy example. This is just a hypothetical made-up data set. Suppose you've done a clinical trial with 600 patients, 300 in treatment A and 300 in treatment B, being the test and the control arms respectively. And you've done this trial and you look at the overall Kaplan-Meier curves, and you'd be disappointed by the, and if you run this through a Cox proportional hazards model and you do an unstratified log rank test, which, as I said, seems to be surprisingly commonly done, you'll be disappointed. You'll get a one-tailed p-value in this made-up data set of 0.105. You're hoping it's less than 0.025, which it is not. And you're hoping, maybe at the start of the trial, you were hoping that the hazard ratio was, at least 0.75 or lower, which it is not. And so you might walk away disillusioned. And if this was a mid-stage clinical trial, then this particular drug, experimental drug, likely would not be advanced further because the results seem to be disappointing. And I'll explain to you in a few minutes that you would have walked away with the incorrect conclusion. And I'll explain why. So what is also done is, you might look at the overall, and then you might say, well, but I pre-specified certain subgroups. And this is very commonly done. let me look at it by these pre-specified subgroups. And then you might say, look, at least the treatment effect was in the right direction. Less than one is better. But it's fairly consistent, and unfortunately, it doesn't seem to be good enough. And so perhaps we should not be investing in this in this experimental treatment for further study. Now, this is just for completeness to point out that if you were to look at the estimated log hazard ratio functions, and then look at evidence for departures from proportional hazards using what's commonly used, this Gram-Thurneau test. There are many different ways to do it. This is one common way to do it. You would pick up evidence of non-pH. And so, okay, so we are here to caution and say, well, don't do this unstratified log rank test, the unstratified Cox pH model, but actually do the risk stratified version. And if you then come up with a similar conclusion, then that's fine. But let's see what happens in this made-up data set. So remember, it's a 5-step procedure. This has to be pre-specified. In step 1, the team, the statisticians, the clinical folks, everyone works together at the study design stage, and you would basically, in that deliberation, you would collectively decide, of all the baseline covariates that you're going to measure, which ones of those are going to have any plausibility of influencing survival time, regardless of treatment assignment. And so in this made-up example, suppose that thought process had resulted in a total of 50 baseline covariates. It's unlikely that you're going to, in practice, in this design stage deliberation, come up with 50, but we wanted to sort of test the limit here. Go add in a whole bunch of noise covariates. Okay, so let's say you come up with 50 at the design stage. These are pre-specified in the analysis plan, if you were to think about doing this for future trials. In step 2, in a pre-specified manner, these 50 covariates, most likely, maybe just a handful of these are really influencing survival times. It's highly unlikely that all 50 of them are doing that. So you really want to weed out the noise covariates. And among different options, we like to use some machine learning tools. So we are big fans of elastic net Cox regression. This elastic net method was developed by Huey Zhou. He was actually my summer intern from Stanford, gosh, maybe 20 years ago. So he and colleagues had developed this. It's a remarkably ingenious idea, and is very popular in the machine learning field. And again, because my time is limited, I won't go into too many technical details, but just point out which tools we are using. And so in this case, we use the ElasticNet tool. We basically run this ElasticNet Cox regression model. And the idea is to really help us through this tool, which of these 50 candidate covariates really ought to be removed from consideration, which ones should remain. So in this made-up example, these 10 covariates survive this filtering step. Some of these are continuous, some are binary. So they've now gotten past step 2. Now, again, the data are still blinded. So the algorithm does not know whether a given subject in the trial got treatment A or treatment B. Okay, the algorithm is still blinded at step 3. Now what we're going to do is to use these surviving covariates at the end of step 2 and put them into a conditional inference-free tool. Again, we did not invent this tree-based approach. This was done by Hawthorne and Roll. We're just using this in this manner to really get ultimately what we want, which is we want to take the overall population, which is a mixture of risk, of patients with very different baseline risk, and to separate them out into high, moderate, low risk patients, subgroups. And that is what we are doing here with treatment blinded data, and we're using this tool to do it. Okay, so visually, then what is this tool going to do? And there's a step 3A and a step 3B. Again, if you're interested in those details, please do take a look at the 5-star paper, which was published back in 2020. So really, when all is said and done, at the end of step 3, what this method, this pre-specified treatment blinder algorithm is revealing is there are 3 very distinct types of patients. These patients shown in red in the bottom panel, they happen to be the one in this hypothetical example where x1, which is a binary covariate, x1 is 0, and there was a continuous covariate, which the algorithm finds an optimal cut point, x26 below 0.35. This combination of x1, 0, x26 less than or equal to 0.35 are the high-risk patients. And similarly, you are seeing the overall pool survival curves for the high, the moderate, and low-risk patients. Clearly, they're well separated. And then in step 4, for each of these formed risk subpopulations, risk strata, so you can see visually S1, these are high-risk patients. You see the survival curves are coming down quite quickly. And in this case, we can estimate for these types of patients, after treatment unblinding, what is the hazard ratio for the moderate risk patients in the middle. For the lower risk patients, what is the hazard ratio? And we can then combine these in step 5 to get overall final inference. Now, how you combine these, again, I don't want to go into the technical details, but suffice it to say that we are taking a weighted average of the estimated log hazard ratios for each of the risk strata. And there are different ways you can do this weighted averaging. And so again, the details are in the paper where we basically use this adaptive weighting. And this actually works remarkably well. And it's an honest approach. It gives you unbiased results. It controls type and error. It has all the good properties that you want in any new procedure. Okay, so this is just to then show you overall. We started out, remember, we started out with, in this made-up example, we started out with the overall unstratified survival curves, and they looked discouraging. The p-value from log rank was discouraging. The hazard ratio estimate itself was clinically uninteresting, as summarized here in the table. And now, when you run the same data to this 5-star approach, remember, it separates out, instead of looking at this directly overall, it first separates out into the low, medium, high risk strata. It then gets an estimate within each of these and then combines. So at the end, you get an overall point estimate, which is interpreted as an average hazard ratio across these risk subpopulations of 0.76. Of course, we would then also be reporting, to explain where the 0.76 came from, we would be reporting the individual hazard ratio that we showed you on the earlier slide. So this is encouraging. This hazard ratio is closer to being clinically meaningful, and the p-value is more encouraging. Now, of course, this is just one made-up data set. One made-up data set doesn't convince us or anybody else. The big question is, does this really work on real data sets? And of course, our paper includes simulation results where we show that this is a very promising approach. It has remarkably robust simulation findings, much more powerful than current practice. But what about real data? So with that in mind, what we did was we said, let's get our hands on some large clinical trials. And because I work at Merck, I've been working at Merck for almost 30 years. So we basically were able to secure the actual data sets from 3 large trials: the Victoria trial, the STRAP-2P-TIMI trial, and the REVEAL trial. These are just acronyms of these large clinical trials. We're showing you here the sample sizes, so large trials. Interestingly, the Victoria trial, they had what we call 6 design strata. So meaning they had pre-identified for the primary analysis, they were going to use a stratified analysis, and they had pre-specified what those stratification factors or stratification covariates were. So it's not like in practice, all the analyses that are reported out there are unstratified. No, in fact, it's fair to say that most of them use stratified, but we showed you earlier in the presentation that surprisingly, even now, even in 2024, we are still seeing in reputable journals, you know, examples where the analysis was unstratified. And here's an example that, and I will discuss this a little more in a few minutes, the REVEAL trial. So what we've done here is we've now retrospectively, for purposes of illustration, we have reanalyzed these data using 5-star, and we've compared the 5-star results to the results that were originally reported in these publications. All of these have been published in the New England Journal of Medicine, one of the top journals in the US. And, you know, we were curious to see, you know, our hunch was that we are going to see treatment effect dilution. Certainly, if there was no stratification in the analysis, for reasons that I'd explained earlier, if you mix up, you know, low, medium, high risk patients, and you just do the analysis directly without separating them out, then theory tells us that you are going to get treatment effect dilution, meaning that the hazard ratio that is being reported using an unstratified analysis is probably incorrectly shrunk closer to 1, which means that you are diluting the reporting of treatment benefit, which is obviously something you don't want to do. Well, you don't want to exaggerate treatment benefit either. And then, of course, in this case, we will see that if there were design strata used, meaning the analysis was stratified, and if you still see, as we see, as I'll show you in the next slide, what appears to be a dilution, then the question is why? Okay, and these are, again, all of you will have copies of these slides, so please don't worry about that. So when you look at these, all the sort of details are given there for completeness. Now let me reveal what we call really the main slide of this presentation. And here it is. Okay, so collectively in these 3 large cardiovascular outcome trials, we looked at the primary endpoint and we looked at the key secondary endpoints. And these are, you know, these are where the endpoints are time to stroke or time to the earlier of cardiovascular death or myocardial infarction, which is a heart attack. So these are most of these are composite endpoints. In some cases, just a single endpoint, such as cardiovascular death. So what you see on the x-axis, on the horizontal axis, is we have a combination of which trial is that data set from and what was the endpoint that we were looking at. On the y-axis, we have the hazard ratio. Now, in the blue line, this line on the top, this is the hazard ratio that was reported in the original publication in the New England Journal of Medicine. And just for visual purposes, we just sort of ordered them from the smallest to the largest, really just for visual purposes. And then simultaneously, we're now showing you for each of these, so for each of these 19 examples, what was the corresponding hazard ratio that we got using a 5-star method. And the result is quite striking, in most cases, with one exception, where we see that the point estimate using 5-star is actually slightly larger than the originally reported hazard ratio estimate. In most cases, we see that the 5-star estimate is further away from 1, meaning less than 1, meaning a stronger treatment effect. So really, this tells us that if you believe that the 5-star method is actually is the more honest method, and not honest is maybe not the best word here, is the more accurate method. And in truth, we've shown you using the toy example, and certainly in the paper if you read it, you will see through simulations, we sort of talk about bias. So 5-star tends to be under the scenario that we've imagined, true risk strata and true hazard ratios in each of these risk strata, which we're averaging to get to the overall truth. Then this is not a surprise to us. You should expect to see, if the trial is unstratified, we will likely be getting hazard ratios that are better, as shown here. And I hope you can still hear me, because my internet, I got a warning.
Yes, we can hear you, Devan.
Thank you, Piali. Okay, so also please note that in this case, in 4 of these 19 examples, as shown here on the bottom, not only are we seeing a notable reduction in the hazard ratio, meaning much more clinically interesting and of benefit to patients, right? In 4 of these examples, what we find is that it actually would have overturned the original conclusion. So the original conclusion was that there was no evidence, no statistical evidence of treatment benefit, because the p-value had not sort of made the threshold. But in this case, in these 4 instances, we see that the p-value is statistically significant. And of course, that's more for completeness. Really, the more important part is, what about the reporting of the treatment effect? Because in clinical practice, when this goes to regulators, when this goes to insurance companies, when doctors and patients look at these results, in either in what's called the drug label, or if some of them actually read the publications, they really want to know, what is the treatment effect? And this is quite striking, that the treatment effect seems to be diluted in these reported publications. I'm going to zoom in just to explain, to give you an example or explanation for why we are getting stronger results here. So let's look at the example. So this first example, this is the REVEAL trial, and we're looking at cardiovascular death. So this was the original analysis that was reported. The p-value, the one-tailed p-value, was nowhere close to 0.025. The hazard ratio estimate was uninteresting. And in the cardiovascular setting, really, hazard ratios, it's not uncommon for these trials to be designed to detect hazard ratios of 0.85, which may seem unimpressive to some people, but remember, cardiovascular disease is very common. And if the drug is approved, hundreds of thousands, maybe millions of people over time are going to be taking these drugs. So even if you see a 15% reduction, that's actually meaningful. So if the hazard ratio, is in the 0.8 to 0.85 range, that is considered clinically meaningful. So the 5-star analysis does reveal that, yes, the overall average hazard ratio is clinically meaningful. The p-value is notably smaller than the original reported p-value, as is the hazard ratio. So what happened here? The original analysis was unstratified. And what we see here is that the 5-star algorithm, this treatment blinded algorithm, it reveals that there are 3 very different types of patients in this trial. The patients that have heart failure are the ones who are dying the fastest. The ones who did not have heart failure on the top and had a glomerular filtration rate, so this is a measure of kidney function, had sort of better kidney function, they tended to have longer survival, overall, and the others were in the middle. So again, if you don't separate out into clinically meaningful risk subpopulations, then you could suffer the consequence of this treatment dilution. Let's go to this another example. This is now from the Victoria trial. So in this trial, again, here's the originally reported result for CV death. Now this trial had used design strata. So at the design stage, they had, based on the available information at the time of the trial being designed, they had surmised, speculated, that perhaps the most important risk factor is region. And so the trial had been the randomization had been stratified by region, and then within North America by Black and non-Black. So the expectation when the trial was designed that at the end of the trial, if you were to look at the pooled survival curves across these different design strata, the expectation was that they would be well separated. But clearly they're not. They're not. I mean, they're lumping pretty closely together. So this is an example where a well-intended team at the design stage with imperfect knowledge, they had placed a bet that yes, they are going to stratify, and they picked these stratification factors, and it turned out that these turned out not to be strongly prognostic for survival. In contrast, if you look what happened with the 5-star algorithm, okay, this is, of course, this is a looking back analysis. So the 5-star algorithm says no, region is not the prognostic factor of interest. It's this marker that is routinely measured in heart failure patients. This was a heart failure patient trial, and there's this measure that's taken at baseline called LT-proBNP. So this is measured routinely in both the diagnosis and the management of heart failure. And also there's this thing called NYHA class. So the higher the class level, the sicker these patients are. So not surprisingly, and higher NT-proBNP numbers mean the patient is more sick. Not surprisingly, the algorithm, which has no knowledge of this clinical feature, it just looks at the data in this treatment-blinded manner, and it says you have these 4 very different types of patients, and they are clearly well separated. And again, if you look at the overall results, the 5-star analysis reveals a hazard ratio estimate that is clinically meaningful and that is notably smaller than the original. So again, this is yet another example of treatment dilution, and you get a very favorable p-value. And again, why is this? Why did we get a better result? Why is there evidence of treatment dilution? Again, because the factors that have been chosen to stratify based on risk turned out not to be prognostic risk factors. So in summary, really, we want to end with this almost now obvious, hopefully, points that risk unstratified survival analysis really should be avoided. It's still happening, not very often, but remarkably common still. And so you might say, okay, well, let's do a risk stratified analysis, and at the design stage, why don't we pre-specify what the risk factors are? Of course, that would be ideal, but what we are finding, having looked at many real data sets now, beyond the ones that I've just shown you, that really at the design stage, there's imperfect knowledge, and to pre-select what the risk factors are that you should adjust for randomization or for the analysis is not as easy as you think. So what you could, instead of trying to agonize over what the right risk factors are, why don't you, in an objective, unbiased manner, allow the data itself to reveal what they are? And that's what this 5-star method is. And I've described it. I should point out that the implementation is very easy. Rachel has developed this amazing R package that you can download from GitHub to implement this for your data. So again, this is just one example of the kind of statistical innovation that the statisticians in pharma do. Certainly at Merck, we are very proud to be part of the community in pharma where this type of innovation is being added, really ultimately for the benefit of the patients. So I'm going to thank you so much for your time and stop here, and let's have Q&A.
Thank you so much, Devan. Very interesting presentation, of course. We have several questions both in the Q&A and in the chat panel. So I'm going to start, not sure if we will have time to cover all of them, but let's see how much we can get those answered. So starting off with this question from Avirup, he says that my question is, since the proposed 5-star approach estimates a weighted average across the different risk strata when finally estimating the risk stratified hazard ratio, don't you think the final risk stratified hazard ratio is likely to suffer from non-collapsibility issues since the risk stratification is leading to targeting of a conditional estimate such as the conditional log hazard ratio?
Thank you. Great question. Yeah, so the importance to the estimand is very important. So if you define the estimand after the risk strata are formed, and so we say, look, before treatment unblinding, right? That's the key. Before treatment unblinding, you say, you know what? Based on the data we've collected, it seems that there are 3 or 4, depending what you get, very distinct types of patients. Here are the heart failure patients with here are heart failure patients, here are patients without heart failure, but who have compromised kidney function. And here are the patients without heart failure and intact kidney function. And once this has been specified, then clinically, the estimate of interest is what is the average hazard ratio across these different types of patients. And so when we've discussed with our clinical colleagues, what they say is that, yes, they really like this because they like to think in terms of, okay, tell me what is the treatment effect in the low risk patients? What is the treatment effect in the moderate risk patients? Treatment effect in the high risk patients? So this methodology gives you that, but in terms of the estimand and the single point estimate that we get, the estimand should be interpreted as the average of the log hazard ratio. So the log is just for convenience. So in the end, of course, people understand hazard ratio. But so on the log hazard ratio scale, this is completely collapsible. So we are taking an average of the log hazard ratio, and of course, you can report an average log hazard ratio, but most people want to convert this back to a hazard ratio, so you're exponentiating it back. So the issue of collapsibility is actually an issue. Back to my teaching example, if you don't risk stratify, then you're creating these non-pH conditions. And what does that hazard ratio mean? Because you are collapsing across different types of all mixed up patients. And that really doesn't make sense. It doesn't make sense to have a number which is sort of the average hazard for treatment A divided by the average hazard for treatment B. That number really is hard to interpret. What you want is the average hazard ratio. You don't want a ratio of average hazard. You want an average of hazard ratios.
Thank you so much, Devan. Moving on to the next question, I thought this is really interesting from industry and drug development perspective. Rajveer is asking that, do you know of any application of this method as of now on any phase 3 pivotal studies and how the agency is taking it?
Yeah, Rajveer, thank you so much for the question. It's so funny because anytime we present new methods like this, the first question the audience asks is, will FDA approve it? Of course, they can only approve it if you ask them. That's step one. So let me answer, Rajvi, this question in 2 ways. So yes, within Merck, we actually have 2 very large cardiovascular outcome trials that are currently being designed. And indeed, for these, 5-star has been pre-specified in the protocol as a sensitivity analysis. So it's not yet the primary analysis, but it's a sensitivity analysis and it's pre-specified. So the question then is, when will it become a primary? It's going to take some time. I should point out that the 5-star approach, which is basically, you can think of it as an approach for covariate adjustment, right? In some sense, it is aligned with 2023 FDA guidance on covariate adjustment in clinical trials. I say that because if you look at their guidance, they ask for 2 things. One is pre-specification, which we have here, and they explicitly mention the word before treatment unblinding. So the algorithm delivers both the things that the FDA guidance is asking for, which is pre-specification of everything in great detail, and that the covariates that are being identified to form the risk strata are being identified using treatment-blinded data. This should not ever be done on treatment-blinded data. Everything falls apart. All the statistical properties fall apart. So a long answer to a very good question that yes, are we making progress with implementation and real trials? Yes. And hopefully someday it will be used as the primary.
Thanks, Devan. Okay, next question. I'll take so two similar questions in the chat. And the question basically says that what is your viewpoint on data driven risk strata identification when the strata are actually clinically meaningless? That is, numerically, they're able to improve power or enforce proportional hazards. But what if the strata are what if the strata are defined by variable combinations that have nothing to do with each other biologically?
Very good question. Very good question. So let me start with the good news. We have applied this to, gosh, at least 25 data sets. In every case, the risk strata that have been identified, we've taken the results back to our clinical colleagues, and they say, yeah, the results make sense, right? Which was a big sigh of relief, because what you don't want is to get results that are hard to interpret. Yes, I completely agree with the person who has the question that if the method is delivering results where the risk strata are head scratchers, like, wow, like why? We would never have imagined this would be a risk factor, right? Now, sometimes, I mean, I remember in one case, one particular covariate was identified as being a composite in the risk identification. And that was a little surprising to the clinical colleagues. But when they looked up the literature, there was ample literature which supported that. But one way to mitigate the risk of having risk strata being formed by covariates that clinically hard to interpret is, remember, you control the covariates that are included in step one, right? In that candidate set in step one. So at the design stage, when you're writing the protocol, you, the statistician and the clinical folks together, you say, look, realistically, which covariates that we are going to measure have clinical plausibility, right? And if at the design stage, if you have stated that, then don't be surprised at the end if you have a risk stratum that's defined by the covariates that you had agreed had clinical plausibility. So yes, that is one way to sort of make sure that the results are not hard to interpret at the end.
Thanks. Okay. Moving back to the Q&A, and here I'm going to combine a couple of questions kind of in the similar line. So the question is there any risk to pre-specifying and including too many possible confounding variables? Or does the elastic net approach limit the impact of including too many covariates? And in a similar line, another person has asked, why did you choose elastic net in particular?
Yeah, great question. So I like elastic net not only because it was, well, not because it was developed by my summer intern, 20 plus years ago. Obviously not. No, we actually have looked at many different approaches, including random forest, elastic net, and other approaches. And what we found is that elastic net, and of course we looked at lasso, we looked at ridge regression. There's so many different approaches you can use. Elastic net turned out to be, in our investigation, the most robust. And not surprisingly, because elastic net really, is a good compromise between ridge regression and lasso. And of course, this is just a side remark, but the reason I came to America many, many years ago from India was to actually study ridge regression. And so the person who invented ridge regression was going to be my advisor at the university when I went to grad school here. And so kind of brought back fond memories of ridge regression. Anyway, so elastic net has has the nice feature, and it is more accommodating of covariates that tend to be more strongly correlated. Lasso is not as robust, right? So that's the beauty of ridge regression. It's more accommodating. But of course, again, going back to step one, be careful when you select these covariates. Do it based on clinical plausibility, and there's no point nominating two candidate covariates that are known to be, almost perfectly or very strongly correlated. I mean, that would not make sense to begin with.
Okay, thanks so much, Devin. We probably just have time for one more question. So I'm going to give the last question from this person as an anonymous attendee, but still. What are the extensions of this method, or have you looked at any extension of this method to non-survival endpoints? Any continuous endpoint, but not on the survival scale?
Yes. And I'm going to make a shameless advertisement for a talk I'll be giving at the Joint Statistical Meetings in Portland this year. So yes, indeed, we have extended this thinking to the analysis of continuous endpoints. I'll be giving a talk in London in September, where I'll be talking about not only about application to survival, but to continuous and binary. So the binary feature is currently being polished up. So again, this is joint work with Rachel. So yes, we've actually finished the work for continuous. That is what we'll be presenting at JSM folks in the US in Portland in August. So yeah, because the concept is quite easy. I mean, in current practice, let's say you have a continuous endpoint and you're interested in a difference in means. Well, what do we do in a clinical trial? We say, we want to compare these two means, and we are going to be, our analysis model will include these covariates. That's pre-specified in the protocol. And we say, why do you want to pre-specify? Why don't you let the data in a treatment-blinded manner identify which truly are the prognostic covariates? And the results are spectacular. I mean, they are, not surprisingly, they show that is much better practice. And it's so easy. It's so easy. You can pre-specify everything. And so again, if you happen to be in Portland, if not, after August, if you want a copy of my slides, please reach out, send me an email. I'll be happy to share that information with you.
Great, thanks so much, Devin. And just one final thing. Will the slides from today's presentation be available for the audience?
Yeah, I'm happy to share the slides.
Okay.
Mel, you're going to arrange that, or Ahia, or?
Yeah, we can definitely, send an email, or you can even put it in the chat, if that is okay with you here as an attachment.
Yeah, I'm not that savvy, so
that's okay. We will take care. Yeah, please send us the slides, and we will definitely take care of that.
Yeah, and of course, if people want, if they didn't have time for questions, please send me an email. I'm more than happy to engage with you in email.
Thank you so much, Devin. I'll let Hia give the concluding remarks. Thanks.
No, thanks a lot. This was the best talk, as always, Devan. Every single time I listen to you, hear to you, it's always a pleasure. And thank you for doing it for IISA. And I want to thank all the participants for today, especially here in the East Coast during their lunchtime and listening to this. It means a lot to us. And there will be the next session in July. We will be reaching out to you with more information. But also don't forget to fill out the feedback form so that we can improve. Again, thank you. Thanks a lot to everyone.
Thank you so much. Bye.
Thanks. Bye-bye.
Thank you. Bye.